When you look at the sheer volume of data generated by modern genetics, it really feels like we should have solved complex diseases by now.
You'd think so.
Yeah, I mean, we've mapped the human genome. We have these massive genome-wide association studies, GWAS, pulling in DNA from literally hundreds of thousands of people.
Huge data sets.
Exactly. And we can point to a specific genetic variant, a single nucleotide polymorphism, and say, okay, people with this specific SNP have a higher risk of Parkinson's.
But then nothing.
Right. The diagnostic landscape just remains totally murky. Have you ever wondered why, with all this incredible data, we still struggle to trace the actual causal chain from a DNA typo to a failing organ?
Well, the bottleneck isn't a lack of data. I mean, it's really the nature of the genome itself.
How so?
So the vast majority of these disease-associated variants uncovered by GWAS, they don't actually sit neatly inside the 1 to 2% of our genome that codes for proteins.
Right. The coding regions.
Yeah. Instead, they fall into the non-coding regions, what we often call the regulatory dark matter.
The dark matter. I love that term. Well, welcome to the deep dive, everyone. Our mission today is to decode this fascinating new computational method designed to basically illuminate that dark matter.
It's a really elegant solution.
It really is. We're looking at a 2025 technical report published in Nature Aging, and it's detailing a tool called scMORE.
Right, which stands for single-cell multiomics regulon enrichment.
Single-cell multiomics regulon enrichment.
Got it.
And its goal is to essentially read the invisible ink of our regulatory DNA.
That's a great way to put it.
So think about it like this. If we think of gene expression as a construction site, GWA might tell us, hey, there's a problem on this site, but it doesn't tell us what the problem actually is.
Exactly. There's a delay, but you don't know who dropped the ball.
Right. So the target gene is the worker building the protein. The regulatory peak, or the enhancer region, is the work order.
But a work order doesn't just execute itself.
Exactly. You need a manager to sign off. And in biology, that manager is a transcription factor, or TF.
Which brings us directly to the core mechanism of the scMORE paper, right? The eRegulon.
The eRegulon. Let's break that down.
So an eRegulon is that entire functional triad. It's the manager transcription factor binding to the regulatory work order to activate the worker gene.
Okay. Manager, work order, worker, all linked up.
Right. And historically, bulk sequencing would just grind up a whole tissue sample and give us this messy average read of these networks.
Like a smoothie, you don't know what the individual fruits are doing.
Exactly, like a smoothie. But scMORE leverages single-cell multiomics. It's analyzing a transcriptome via scRNA-seq.
That's the active genes, right?
Yeah, the genes that are firing. And it's also looking at chromatin accessibility via CADAC-seq, which is the open DNA regions. And it does this simultaneously within the exact same individual cell.
Wow. So we can see which genes are firing and which regulatory regions are physically open, cell by microscopic cell.
Right. But building that network from the data is, well, it's tricky.
I want to dig into that, actually. How does scRNA construct these networks? Because the paper mentions building a global TF-mediated gene regulatory network.
GRN, yeah.
Right. And they use a generalized linear model looking at a 100 kilobase window around a gene's transcription start site.
Yep, that's the default window.
But okay, with the notorious sparsity and noise of single-cell data, because I know single-cell data is super noisy, how does the algorithm statistically prove that the manager, the work order, and the worker are actually linked?
Like, how do we know they aren't just randomly firing at the same time?
Exactly. How do we know it's a real causal chain?
Well, the generalized linear model is essentially hunting for robust correlations across thousands of individual cells.
Okay.
It evaluates whether the expression level of the transcription factor, combined with the physical accessibility of the enhancer peak, reliably predicts the expression of the target gene.
So it's looking for a consistent pattern across the population of cells.
Right. It fits a mathematical model to the data. And if the variance is explained by that relationship, it establishes an edge or a link in the network.
But wait, you still have the issue of single-cell dropout, right? When you sequence at a single-cell resolution, you just miss things.
Oh, absolutely. The sparsity is a massive challenge.
And that sparsity makes it incredibly difficult to determine cell type specificity, or CTS, doesn't it?
It does. Because think about it. A transcription factor might be universally expressed in the body, but it might only act as a rogue manager driving disease in one highly specific subset of cells.
Right.
If you try to calculate that specificity by just averaging the expression magnitude across cells.
The dropout noise just washes the signal completely away.
Exactly. Which is why the scMORE algorithm does something really clever. It abandons magnitude entirely and uses cosine similarity.
Okay, I love this mathematical pivot. I remember cosine similarity from high school trig, but what does it have to do with cells?
Well, instead of measuring the volume of expression, which fluctuates based on, how deeply you sequence a cell.
Which is just technical noise.
Right. Cosine similarity looks at the high-dimensional alignment.
High-dimensional alignment. Break that down for me.
Sure. If you plot the expression of our manager across 1,000 cells as one vector.
Like an arrow on a graph.
Exactly.
Right.
You plot an ideal cell type marker as another vector. Cosine similarity measures the angle between them.
Ah, I see. So an angle of 0 means perfect alignment.
Right. It means they fire in the exact same cellular contexts. And it doesn't matter if one cell was sequenced twice as deeply as another because the length of the arrow changes, but the angle stays exactly the same.
That is brilliant. It's completely immune to that dropout noise.
It isolates the pure biological pattern. It gives every node in our eRegulon network a reliable cell type specificity score.
So we aren't just finding a manager, we're mathematically proving their exact jurisdiction.
Exactly.
Okay, let's anchor this real quick before we layer on the genetics. We're taking the dark matter of GWAs and essentially mapping a construction site using single-cell multiomics.
Right, our first big takeaway.
Yep. So bullet 1, GWAS finds disease SNPs, mostly in non-coding regions. Bullet 2, multiomics maps both gene expression and DNA accessibility. And bullet 3, scMORE finds eRegulons, those TF networks, and scores their cell type specificity using cosine similarity.
Which is a huge leap forward.
It really is. So, to you, the listener, here's a question. If you were designing a drug, would you rather target a worker gene or the manager TF that controls the whole site?
Well, targeting the manager requires us to link these pristine cellular networks back to the messy real-world GWAS data, right?
Right. We have to prove the network is actually driving human disease.
Exactly. So to do that, the algorithm calculates a genetic relevance score, the GRS.
Okay, step 3 of scMORE, the GRS, the formula for this in the paper is super elegant. It's PR times PS times GS.
Right. Peak relevance multiplied by peak strength multiplied by gene score.
Let's unpack the mechanics of that multiplication. What's PR?
So peak relevance evaluates the regulatory work order. It asks, what is the highest statistical significance of a GWAS disease variant located within this specific enhancer peak?
So does this peak have a known disease typo in it?
Exactly. Then peak strength measures the biological coupling.
How so?
It asks, how strongly does the physical openness of that peak correlate with the target gene actually firing?
Okay. So even if there's a severe GWAS hit on the work order, if that work order doesn't actually influence the worker gene,
the peak strength zeroes out the risk.
it demands functional impact. That makes so much sense.
Right. And then the gene score, which the paper calculates using external tools like MedGMA.
Like GMA, right.
Yeah, that assesses the target gene's overall population level association with the disease. Multiplying all three together anchors the single-cell network to real clinical risk.
Okay, but here's where it gets really interesting. How does scMORE balance that genetic data with the single-cell specificity data?
That's the trait-associated Regulon score, or TRS. That's the ultimate metric here.
The TRS. The formula is TRS equals GRS plus CTS minus the standard deviation between GRS and CTS.
Yeah, that subtraction part is key.
When I first read this, the subtraction of the standard deviation totally jumped out at me.
Why is that?
Well, if you just add genetic risk and cell specificity together, a massive genetic signal could easily mask a really low cell specificity score.
Right, you'd end up chasing a pathway that isn't actually driving disease in the tissue you're looking at.
Exactly. It's like, imagine grading a student on math and history. Math is our GRS, history is our CTS.
Okay, I follow.
You don't want a student who has an A+ in math and an F in history, right? You want the student who gets an A in both.
Right, the well-rounded student.
Yes. So the standard deviation acts as a severe variance penalty. It essentially punishes discordance.
Yeah.
You want a Regulon that is a powerful driver of genetic risk and highly specific to the cell type.
Because if a network scores in the 99th percentile for genetics, but only like the 50th for cell specificity, the variance is huge.
And the penalty drags the overall TRS down. It demands biological congruence.
It does, but any computational model can hallucinate a high score if you torture the data long enough.
Oh, sure. So how do they prove these TRS rankings aren't just statistical artifacts?
Through really rigorous permutation testing. For every candidate eRegulon, scHOB runs Monte Carlo simulations.
Monte Carlo simulations,
And
generating matched random controls, right?
Exactly. It generates 1,000 matched random networks. It shuffles the data while preserving the underlying structure, calculates the scores for all 1,000 random iterations, and creates a null distribution.
Wow. So it forces the real network to compete against 1,000 fakes to generate an empirical p-value?
Yeah, it's computationally expensive, but it's the only way to guarantee the signal is actually real.
It ensures that when scMORE flags an eRegulon, we are absolutely looking at a biologically active, genetically risky network operating in a specific cell.
And the researchers didn't just stop at the math. They immediately put this to the test on real clinical data.
Which brings us to our second mini recap, at the 10-minute mark here.
Let's do it.
Bullet 1. GRS calculates the genetic disease risk of a pathway. Bullet 2. TRS combines that genetic risk with cell specificity. Bullet 3. A variance penalty ensures both metrics are strong.
And you weed out the false positives.
Exactly. So, listener, think about this. How often do you think medical research finds a generic genetic risk but completely fails to identify the exact cell type where it's causing chaos?
Probably more often than we'd like to admit.
Right. So, back to the clinical data. Their proof of concept was looking at lymphocyte counts in human peripheral blood mononuclear cells, or PBMCs.
Yeah, standard human blood samples.
And what did the math find?
It identified 4 specific eRegulons managed by the transcription factors ZEB1, IKZF1, FOXO1, and ZNF721.
That's a mouthful.
It is.
Yeah.
But the crucial part is the algorithm claimed these were specifically associated with lymphocyte traits in CD8 plus T cells and explicitly not in monocytes.
And does the known biology back that up?
It confirms it beautifully. Take FOXO1, for example. It is fundamentally required for the generation and maintenance of memory T cells.
The immune system's long-term memory hard drive.
Exactly. And IKZF1 is a critical regulator of hematopoiesis, which is blood cell creation.
Wow. So the algorithm mathematically isolated the actual biological managers driving T-cell function, and it did it completely blind, no prior biological input.
None at all. Just the math and the raw data.
That is incredible. Knowing it can accurately blind identify known immune regulators like FOXO1 really sets the stage for where the paper goes next.
Right, which is autoimmune disease.
Yes. They analyzed single-cell data across 5 major autoimmune diseases: IBD, primary biliary cholangitis, rheumatoid arthritis, SLE, and ulcerative colitis.
But they introduced a really critical new dimension here because, cells aren't static.
Right.
They mature, they react, they change states over time.
Exactly. Capturing a cell at a single moment is like taking a photograph of a car on a highway, right? You know where it is, but you don't know where it's going or how fast it's traveling.
That's a perfect analogy. So to solve that, the researchers used a computational tool called Slingshot to map what's called pseudotime.
Pseudotime, how does that work?
Slingshot basically takes single-cell transcriptomic data and orders the cells along a continuous developmental trajectory.
Okay.
It infers the lineage, tracking a cell as it transitions from, say, a generic precursor state all the way to a highly specialized mature state.
So it's essentially animating the photograph.
Exactly. And when they mapped the autoimmune eRegulons across this pseudotime, they discovered that these regulatory networks are highly dynamic.
Meaning the causal managers actually shift as the cell ages?
Yes. The contrast they found between inflammatory bowel disease and rheumatoid arthritis in monocytes is just a perfect example of this.
So what does this all mean for treating something like IBD versus RA?
It fundamentally changes the therapeutic paradigm. In IBD, scMORE identified an eRegulon managed by HES4.
HES4, okay.
But HES4 only becomes a rogue manager driving disease when the monocyte reaches a specific late-stage state.
A late stage. What's happening in that stage?
That's when the cell is characterized by the massive production of inflammatory cytokines.
Wow. Whereas in rheumatoid arthritis?
In RA, a completely different manager, KLF4, is driving the disease. And unlike HES4, KLF4 is constitutively active across the entire developmental trajectory of the monocyte.
So it's driving general leukocyte proliferation from start to finish.
Exactly.
That is mind-blowing. Two completely different autoimmune diseases leveraging completely different transcription factor managers within the exact same lineage of immune cells, but operating on entirely different temporal schedules.
Right. It makes you reconsider the timing of every single drug trial out there.
Seriously, if a therapeutic is designed to inhibit HES4 for IBD, but it's administered when the patient's monocyte population hasn't reached that specific mature state,
the drug will look like a total failure.
Right. Even though it's the right drug, it's the wrong time. It's not just about the right drug for the right cell. It's about hitting the exact microsecond of its developmental trajectory.
That temporal precision is vital.
Time for recap number 3. We are flying through this.
Let's hear it.
Bullet 1, scMORE proved itself by finding known immune-regulating TFs like FOXO1 in T cells. Bullet 2, it can track disease pathways across changing cell states over time using pseudotime. And bullet 3, different autoimmune diseases leverage completely different TF managers in the exact same immune cells.
Spot on.
So, listener, if a disease changes its manager as a cell ages or changes state, how does that alter the timing of when you should administer a drug?
It's the million-dollar question in pharma right now.
Truly. Now, let's pivot to segment 4, because the researchers push this even further by applying scMORE to one of the most genetically complex and honestly inaccessible environments we have.
The human brain.
Specifically focusing on Parkinson's disease in the midbrain.
Right. They pulled single-cell multiomics data from the human midbrain, which is profoundly impacted by Parkinson's.
And mapping the dark matter in neurology is notoriously difficult. But scMORE identified 77 aging-relevant eRegulons across 7 distinct types of brain cells.
But what really stood out wasn't just the sheer volume of implicated networks, it was the sex-dependent dysregulation they uncovered.
Wait, really? Sex-dependent?
Yeah.
That detail shifts the whole landscape. I mean, we know Parkinson's presents differently and progresses at different rates in men compared to women.
Right.
But tracing the actual molecular causality of that has been completely elusive.
Until now. By comparing neurons from Parkinson's patients to both young and aged healthy control groups, scHOB revealed that these master regulatory switches break down differently based on biological sex.
So the eRegulon activity driving the pathology in male midbrain cells diverged significantly from the female pathology.
Significantly, yes.
We're looking at the invisible ink of genetic risk inside a specific midbrain neuron, operating differently depending on sex. The implications for stratifying patients in clinical trials are just massive.
They are. But, studying the human brain brings up an obvious physical limitation.
Yeah, you can't exactly routinely sample living human midbrain tissue to test these pathways.
No, you definitely can't. Which leads us to the massive secondary resource published alongside this method. If we connect this to the bigger picture, we have scHOB.
scHOB, the single-cell human organoid bank.
Exactly. Organoids are the key here.
For those who might not be familiar, organoids are essentially miniature lab-grown versions of human organs, right?
Right. They provide the complex three-dimensional tissue architecture that flat 2D cell cultures just lack.
And they retain human genetic fidelity.
Yes. So scHOB curates over 1.5 million cells from 10 different types of organoids.
1.5 million cells. That's a massive data set.
It is. And when the researchers ran scMORE on cerebral or brain organoids integrating GWAS data for psychiatric conditions, they successfully mapped cell-type-specific eRegulons to 8 different psychiatric disorders.
That bridges such a crucial gap. It proves we can take population-level psychiatric GWAS signals,
which historically have been incredibly difficult to map to physical pathology.
Right, and we can anchor them to highly specific regulatory networks within lab-grown human brain tissue.
It's basically a sandbox for precision medicine.
I love that. A sandbox.
Yeah.
Because if you can identify the exact rogue manager in a cerebral organoid that mirrors a patient's psychiatric genetic profile, you can screen thousands of compounds against that specific cellular state.
Without ever touching the patient.
Exactly. But, okay, let's ground this for a second. While the potential is staggering, we have to look closely at the limitations and the evidence grading of scMORE.
We do. It is fundamentally a hypothesis-generating engine.
Right. It establishes rigorous mathematical associations, but not absolute biological causality.
Exactly. It hands you a highly detailed map and says, dig here.
Dig here. But you still have to pick up the shovel.
Right. You still have to conduct the downstream clinical validation.
Like CRISPR knockouts or functional assays.
Exactly. To prove that physically disrupting that manager actually halts the disease in a living system.
And what about the computational constraints?
Well, the model is constrained by its default parameters. Remember earlier we mentioned it maps GWAS SSMPs to genes within a 10 kilobase window, and it links transcription factors to targets within a 100 kilobase window.
Right, those are the defaults.
Now that captures a massive amount of biology.
Yeah.
But regulatory elements can occasionally physically loop in 3D space and act across vastly longer genomic distances.
Ah, I see. So the algorithm allows researchers to swap in different mapping strategies or long-read sequencing data.
Yes, it's flexible. But those default boundaries mean some really distal, faraway regulatory interactions might just slip through the net initially.
That's vital context. We aren't capturing the entirety of the genome's complexity just yet, but we are bringing an unprecedented level of focus to the networks we can see.
Absolutely. It's a huge step forward.
Well, we started this deep dive, mired in the dark matter of GW, staring at genetic risks without knowing the biological mechanism.
And we've seen how scMORE cuts through that single cell noise.
Right, using the geometry of cosine similarity. We've explored the variance penalty that ensures our targets are both genetically lethal and cell-specific.
We've tracked rogue immune managers shifting their tactics across pseudotime in autoimmune diseases.
Mapped sex-specific breakdowns in Parkinson's and projected complex psychiatric risks onto millions of organoid cells.
It really provides a mechanistic framework that finally allows us to interpret population genetics through the lens of single-cell biology.
It completely redefines what a diagnosis actually looks like at the molecular level. So as we wrap up today, I want to leave you, the listener, with a final thought to turn over.
It's a big one.
It is. If computational methods like scMORE can pinpoint the precise molecular manager operating in a highly specific cellular state driving a complex disease like Parkinson's or rheumatoid arthritis, are we approaching a threshold where we stop conceptualizing treatments by the organ they target?
Like brain drugs or gut drugs?
Exactly. Will the future of medicine bypass that entirely and instead focus purely on deploying an intervention to intercept a specific transcription factor in a specific microscopic lineage precisely when it attempts to change states?
What does that mean for the architecture of tomorrow's clinical trials?
That is the question. Keep questioning, keep exploring, and we'll see you on the next deep dive.